Okay, so now let's take a look at our very specific host defense mechanisms. In the last chapter, we discussed non-specific, which again meant that our first and second line of defense are not specific to any certain type of pathogen. They're not specifically looking out for the influenza virus or streptococcus, that causes strep throat or whatever. They're again just trying to keep any and everything out, something that doesn't belong in our body. Our first and second line of defense are designed to just keep everything out. Now, in this chapter, we are going to take a look at our specific lines of defense, which is our immune system. And this is going to mean that we have either been, been exposed to the pathogen before, either through, naturally or through a vaccine of some sort. We're going to take a look at different ways that our immune system works. So let's take a look now at chapter 16. Okay, so chapter 16, our specific host defense mechanisms. So immunology is what we're looking at in this chapter. So we'll start with an introduction. We'll take a look at what immunology is and understanding that. Primary functions of the immune system. So what all is involved in the immune system? We'll take a look at major arms of the immune system. You guys may be familiar with cell-mediated immunity versus humoral immunity. And we're going to be real brief with that. And I'll show you guys what I want you guys to be familiar with on that part. We'll take a look at those major arms of the immune system. Immunity, different cells that are involved in immunity in the immune system, where those immune responses occur. Again, we'll briefly discuss at the beginning and then a little bit later too, the difference between the two, humoral versus cell-mediated. And then we'll end with hypersensitivity. I'm going to be real brief with hypersensitivity and autoimmune diseases. We really don't get into immunosuppression in the immunology laboratory, so we'll skip through that if I haven't already taken it off the slide already. Okay, so immunology. So as we are familiar with already from anatomy, our immune system is our body's defense system. So immunology is the specific or the scientific study of that system, of that specific system, the immune system. The primary functions of the immune system are to do this. One, to differentiate between self versus non-self. So again, what belongs in our body and what does not. And then on the other hand, the things that do not belong, our immune system is to destroy that. So the job of the immune system is to distinguish between the two of them and then destroy the things that don't belong. So that's essentially what our immune system does. We have two major arms of the immune system. We have what's called humoral immunity versus cell-mediated immunity. And I'm going to be just real brief as possible with this because we could get really complex with humoral versus cell-mediated, but I'll explain to you guys what I want you guys to be familiar with. Humoral immunity is the involvement of antibodies. So we've heard the term antibodies very often, that antibodies are created in response to antigens that enter into our body. Like I said, antigens is kind of a synonym for pathogens. Antigens means something foreign, so something that doesn't belong, so non-self, enters into our body. Our body creates antibodies in response to those. That is the humoral immunity arm of the immune system at work. So that's what happens here. So it gives kind of a specific definition. This is where special glycoproteins in our body called antibodies are produced by cells that we have in our body called B cells, and those are going to work to destroy those specific microbes. So humoral immunity, the main thing that will do is the involvement of those antibodies. Whereas cell-mediated, just as the name implies, that word cell in it, this is going to be the involvement of a variety of cell types that we'll take a look at. Antibodies really play a very minor role, if any whatsoever. So that's what we're going to take a look at as far as the difference between the two. Remember, antibodies are involved in our humoral immunity, and then we'll take a look at different types of cells involved in cell-mediated, so not antibodies. Okay. So again, real brief, this just shows a breakdown of the two. Humoral immunity involves the production of antibodies, and antibodies are going to play that major role in that part of the immune system. Whereas cell-mediated, so like I said, a variety of different types of cells are involved here. Macrophages, which are just large versions of monocytes, essentially, which are phagocytic cells. Remember our phagocytes that we talked about, part of the second line of defense. But macrophages are our largest version of those phagocytic cells, and they're part of our cell-mediated immunity. So just some other examples, we have T cells, we have T helper cells, cytotoxic T cells, and I'm going to break down a couple of these for you guys, but I'm not going to have you guys know all of these. As again, you can see from this list here, it is a variety of different types of cells. Delayed hypersensitivity T cells, natural killer cells, we'll define those just as the name implies there, what they're going to do. Killer cells and granulocytes. Antibodies may play a role in some types of our cell-mediated responses, but they really don't play a major role. So humoral antibodies, cell-mediated, different cells. Those are the two things that, the two types of immune systems that, I mean, I'm sorry, not immune systems, but arms of the immune system that we have that are involved in our defenses when it comes to our immune system. So immunity itself can really be described as what's called acquired immunity. As I mentioned at the very beginning, we really have to be exposed to a pathogen in some way in order for our body to be prepared for how to fight it. We don't, for something that's brand new, the very first time we've ever been exposed to something, our body has no idea what it is. So it has to teach itself what that particular pathogen is in order to be able to fight it off. So the first time we're exposed to something, we're more than likely going to get sick from it because of the fact our body doesn't know how to fight it. But over time, the more that we're exposed to things, whether it's naturally or through vaccines, when we're exposed to things, we learn, we're essentially teaching our body what these viruses and bacteria and fungi, what these guys look like, so that we know how to fight them off better the next time. So that's what I mean by immunity. Acquired immunity means immunity that results from the active production or receipt of antibodies during our lifetime. So again, antigens are those foreign materials, those invaders that make its way into our body, and then we create antibodies in response to those. So we ourselves create those antibodies, but sometimes we have to receive those antibodies. So that's what we're going to take a look at today. So we're going to see the difference between we ourselves producing the antibodies. That's where you're going to see the term active. And then if we have to receive the antibodies, so in other words, if they're being passed on to us, that's going to be called passive. So that's the definition that we're seeing there. Active acquired immunity means that antibodies are produced within the person, so within ourselves. Usually this is going to provide a much longer lasting protection because again, in an example where maybe I've been exposed to, say, the flu virus, I create antibodies in response to it, which means the next time I'm exposed to that virus, well, with the flu, as long as it's within that same year, because again, the flu changes from year to year, as long as I'm exposed to it that same year, I recognize it. My body produces the antibodies again because it remembers creating those the first time. So that's usually going to provide a little longer lasting protection. Whereas passive, in this case, the antibodies are received that were either produced by another person, so an immune person that has the antibodies already, they create some type of serum that can be passed on to other individuals or even an animal. So they create something that can be passed on. So this is typically going to only provide a temporary protection because we ourselves did not create those antibodies. Somebody else had to create those for us. So this is usually to make sure that at this particular given period of time, I don't get sick from it. But again, if I'm exposed to it later, I may. So that's the difference between active and passive. And we're going to break this down even further. And hopefully you guys remember this from anatomy. So there's actually two types of active immunity and two types of passive. And that's where we're going to see the terms natural and artificial come in. So when we use, again, the active acquired immunity, remember that means we ourselves are creating our own antibodies. So when we ourselves create these antibodies, like I said, this is going to be a little bit longer lasting, but it's because we are being exposed to the antigens themselves. So we ourselves have been exposed and we are teaching our body how to respond, essentially, how to respond to these particular pathogens. So that's what we're going to take a look at here. So there's natural, so meaning that just as the name implies, it occurs naturally, or artificial. So this is where those vaccines are going to come in. So that's what I mean by artificially induced. That means that because that's what a vaccine is. It's not a medication of any sort. A vaccine is really giving us the virus or bacteria or whatever it is that, we're trying to defend against. It's artificially, we are giving a person, it's typically a weakened version or sometimes a killed off version of that virus or of that pathogen. And so it still teaches our body what that pathogen looks like, but it can't make us sick because it's not active, if that makes sense, or it's not alive. So it's not going to, cause any harm to us, but it still teaches our body how to create the antibodies. So let's take a look and let me show you what I mean. So again, artificial active results when a person receives a vaccine. So a vaccine is that material that can artificially induce that immunity to that infectious disease following that injection or ingestion of that vaccine, because that's what a vaccine is. Most vaccines are made up of either living, sometimes living, depending on the pathogen, or dead pathogens or the toxins that they produce. And that's what we're going to talk about on the next slide. These are examples of the different types of vaccines that are available to us. And I'm going to give you guys just kind of what I would focus on these, and I'll show you what I mean. Okay. So in this case, we see vaccines, again, are stimulating the recipient's immune response to make those protective antibodies. So from that receipt of the vaccine, we are creating these antibodies to this particular pathogen. So there's a couple different types. So attenuated vaccines. So in these particular vaccines, I'm trying to, myself trying to kind of, there's a couple that I really want you to focus on. But attenuated vaccines, this is really just kind of an avirulent strain of a particular pathogen. So virulent, again, meaning that it's very pathogenic, but we can take the same strain of a pathogen and take the avirulent. So again, it's not going to make us sick, but it still introduces that particular virus to us. So it's the non-pathogenic strain of that pathogen. Whereas an inactivated, you can tell just based on the name itself, inactivated, meaning it is a killed off version of that pathogen. So again, upon injection of this vaccine, we are not going to get sick from it because it's not alive. It's not going to be able to spread. But again, it still shows us what it looks like so we can create antibodies for it. A subunit vaccine, so maybe you can tell by that name as well, it's only just a part of that pathogen. What we can do is not what we can do, but scientists who have figured out this type of vaccine can still work, is we can take the antigenic portion of a pathogen, which again is the antibody stimulating portion of that pathogen, but not the whole thing. So it's just a part of the pathogen. Conjugate vaccines, these are a little bit more complex. That these are created by taking bacterial antigens. It's kind of hard to explain, capsular antigens that they themselves are not antigenic, but it can still stimulate the immune system to produce antibodies. So essentially, conjugate vaccines are really just a pathogen that has been kind of reworked. And so it's not going to, again, make us sick. I wouldn't worry too much about that one. So again, the ones I'd focus on so far, inactivated, which are the dead versions of a vaccine. A subunit, which is just a part of that vaccine. And then toxoid. So remember on the definition on the previous slide, it talked about how it's either a live, it can be a dead version of a pathogen, or sometimes it's just the toxins that they produced. And that's what a toxoid vaccine is. It's just the toxins that are produced by the pathogens, what is essentially going to make an individual sick. But without the pathogen itself, it's not going to work. It's not going to be able to make us sick, but it still can introduce to our body what we need to look for when we need to create these antibodies for defense. So again, that's what a vaccine is. It's just and again, all these different vaccines have been created in a laboratory, and you can see there's so many different ones. There's, we've got the antigenic portion, the avirulent portion of a vaccine. We've got inactivated, which is a killed off version of a pathogen. Subunit, which is just the, a part of the pathogen. Conjugate, which has kind of been a bacteria that's been kind of reworked so that our body recognizes it, but it's not going to make us sick. Toxoid are just the toxins produced by these pathogens. So through years and years of studies, we've been able to see that these parts of these antigens can be injected into a patient's body, teaches that patient's body how to respond to it, so that upon the time we maybe are exposed to a live version of that pathogen, we know exactly how to respond, or that patient's body knows exactly how to respond. And that's what a vaccine is. So it's part of that artificial active. So remember, I'm going back to the previous slide for just a second. The difference was natural active. So again, I meant that it occurs naturally. And to give you an example of what I mean by that occurring naturally, that means, again, that I have sat next to somebody that has the flu, they cough on me, I get the flu. That's what I mean by it occurs naturally, that I am probably going to get sick, but at that time where I'm, sick, my body is learning what this particular virus or bacteria looks like, and I'm using the flu, so this virus, what it looks like, so that, again, the next time I'm exposed to it, I know exactly how to respond to it. So that's natural active. And then artificial active is through a vaccine. Okay, there's your different vaccines. So let's take a look a little bit more into passive acquired immunity. So again, passive meant that we had to be passed on the antibodies. We ourselves cannot create the antibodies for this for some reason or another, so they're passed on to us. So remember, this is going to provide only temporary protection. So there's two types. We have natural passive, which in this case means that there's really a couple different examples for natural passive. So it occurs naturally. It's something that's normal, it's natural, but again, the antibodies are being passed on. Great example would be through a mother's blood from a placenta to a fetus. So in the womb. So in the womb, a mother is passing on small antibodies. You don't have to specifically know immunoglobulin G. That's present in the mother's blood are going to cross the placenta into the fetus. Another example would be also through breastfeeding. So another example, mother-baby, is natural passive, is a way to think about that. Because again, through breast milk, mothers can produce antibodies in the event that the baby needs them at that time, because again, a baby has not produced an immune system quite yet. They're still developing that immune system because they have not been exposed to anything yet. So mother can pass on through breast milk. And then artificial passive, again, being passed on these antibodies, but artificially. In this case, antibodies are created in a laboratory. So from an immune person or animal, are then transmitted to a susceptible person that cannot create their antibodies. So somebody who is immune to something, a serum is created from their blood that's able to then be passed on to other individuals. So remember the difference between natural active and artificial active. So again, natural, just probably getting the illness itself. Artificial active through a vaccine. And then passive, so in this case, we have to be passed on the antibodies. Natural passive is through mother and baby, whether it's through the placenta to the fetus in the womb or through breastfeeding. And then artificial passive is just antibodies being passed on through a serum of some sort. So those are the different types of immunity that we could have. So let's look a little bit further into humoral immunity. So again, humoral immunity is going to be the response that our body, produces these antibodies. So again, antigens are your foreign organic substances that are large enough to stimulate the production of antibodies. Antigens, remember, just think something foreign. So something capable of stimulating antibodies are said to be antigenic. So a bacterial cell that has many different molecules, so antigenic determinants is what that would be called, on its surface are capable of stimulating the production of antibodies, for example. So our antigens are what's stimulating our antibodies. So antibodies then are proteins that are created by our lymphocytes, so a specific type of white blood cell in our body, in response to the presence of that antigen. So they are in a class of plasma proteins called immunoglobulins. Not that you have to worry too much about that, those globular proteins in the blood that participate in those immune reactions. So antibodies, what they're going to do, so the amount and type of antibodies that are produced by a given antigenic stimulation really depend on a couple different things. Depends on the nature of that antigen, so meaning just the pathogen itself. The site of the antigenic stimulus, so where is this taking place in the body? The amount of antigen. So again, if we receive more pathogens, we're probably going to create more antibodies in response to it. An example would be, again, I think I've mentioned this before, but indirect contact sometimes only exposes us to a few pathogens. It's still enough to make us sick. But, you know, if somebody coughs on their hand, touches a doorknob, and then I touch the doorknob behind them, I may only pick up a couple pathogens that way. Whereas if somebody coughs directly in my face, I may now have received more pathogens than the other example. So it depends on the amount of antigens received and the number of times the person is exposed to the antigen. The more we are exposed to something, the easier it is going to be for us to fight something off because our body recognizes it. And every single time we are exposed to something, we produce more antibodies in response each time because we say, hey, I've seen this before. I know exactly what antibodies I need to create. And we create those, you know, faster and more so. So what I mean by, again, the person exposed to a pathogen, so the primary response is the initial immune response to an antigen. The first time we are ever exposed to something, as I mentioned previously, we're probably going to get sick from it because, again, my body goes, I've never seen this before. I have no idea what it is. So we're probably going to get sick. It takes usually about 10 to 14 days for antibodies to be produced in that example, again, depending on the specific antigen. So that's, like I said, our primary response. But then following a second exposure to a particular pathogen, this is going to be called the secondary response. And in that case, there's an increased production of antibodies because our body does recognize it and we know exactly what to do. So again, the increased production of antibodies following that second exposure, this is going to be our secondary response. So again, the more we're exposed to something, the more we know what to do, how to respond. In cell-mediated immunity, like I said, there's lots of different types of cells, and I'm only going to have you guys remember just a couple of these. So antibodies are unable to enter cells in cell-mediated immunity. So in this case, it's a complex system of interactions among many different types of cells and the cellular secretions called cytokines. An arm of the immune system capable of controlling these chronic infections by these intracellular pathogens, such as certain bacteria, protozoa, fungi, viruses, etc. So a couple different examples of the cells that participate, as we saw in that chart earlier, we've got your macrophages, helper T cells, cytotoxic T cells, you got your natural killer cells, granulocytes, lots of different examples. Again, I'm only going to have you guys be familiar with a couple of these. So again, the big thing to remember with cell-mediated immunity is that this is the involvement of not antibodies, but lots of other different types of cells. So one example, I like this one only because the name implies exactly what it does. Natural killer cells do just that. Natural killer cells are a subpopulation of our lymphocytes called large granular lymphocytes. Not that you have to know that they resemble lymphocytes but lack the T and B cell markers. I'm trying to think of what I really want you guys to be familiar with on this. The big thing really to remember is that these natural killer cells do just that. They're involved in naturally killing and targeting. So you actually more so this last bullet point here. Natural killer cells are going to kill target cells, which include any type of foreign cell, host cells that have been infected by viruses or bacteria, and then even tumor cells. So big thing to remember here, natural killer cells, know this last bullet point here. That's a big part to remember. That's what they're doing, and know that they're involved in this cell-mediated immunity. Okay, so again, I know that there were lots of other types of cells involved in that. Knowing that those, you know, that there's lots of different types of cells, but I'm not going to have you guys define all those other types of cells. So be familiar with just lots of cells are involved in cell-mediated immunity, but the one that I want you guys to really be familiar with are the natural killer cells. How about that? I think that makes it a little bit easier. All right, let's get into what's called hypersensitivity. So in finishing up and talking about our immune system again is a response to pathogens, right? Our body is being infected by these antigens, these foreign invaders, and we are responding to that. Hypersensitivity refers to when we maybe overly respond. So it's an overly sensitive immune system. There's different types of hypersensitivity reactions. We actually have 4 different types. The first 3 are what's called the immediate type. So this means that it occurs within a few minutes to 24 hours. So it occurs very quickly. So this is whenever we have contact with a particular antigen quickly, either types 1, 2, or 3, hypersensitivity reactions take place. We're going to get into some examples, but the best example of a hypersensitive immune system or hypersensitivity reaction is an allergic reaction. And I'm sure all of you guys have either experienced an allergy before or maybe know somebody who experiences allergies. We know that immediately upon interaction with what we are allergic to, we either immediately respond or sometimes maybe even a couple hours. But that's going to fall into the type 1 category, and it's really going to be the only hypersensitive reaction that I want you guys to be familiar with. But it is an immediate type, so it falls within that category. We'll talk a little bit more about that. And then the delayed type, the only thing I want you guys to know about delayed means that it takes more than 24 hours to manifest itself, and then that's the type 4 hypersensitivity reaction. So of these 4 types, again, know that types 1 through 3 are immediate, so they happen immediately upon contact with an antigen. So again, if somebody is like allergic to peanuts, more than likely they're going to respond right away when they ingest or contact a peanut, depending on the severity of their allergy. And then type 4 is called delayed type, which means that it takes more than 24 hours for your immune system to respond. And when I say an overly sensitive immune system, again, as we are familiar with an allergy response, when we think of hypersensitivity, what happens is our body is creating a ton of histamine in response to this allergy. And it's almost like, oh, hey, I'm super allergic to that. I really need to, do something. And our body just goes out of whack almost. That again, we're creating all kinds of other symptoms from this. That we are, anybody who's again experienced allergies knows that we see a lot of, red noses, swelling. We see a lot of sniffling if we're allergic to pollen and things of that nature. If it's a food allergy, we see some gastrointestinal reactions like those that are lactose intolerant. If it's something like peanuts, we may see an anaphylactic reaction. If we see a very sensitive allergy, just depending, like I said, on the severity of that allergy. But that's what I mean by a hypersensitive reaction. So like I said, the type 1 hypersensitive reaction are also known as anaphylactic reactions, which include your classic allergic responses, such as hay fever symptoms, asthma, hives, gastrointestinals that result from food allergies. Like I said, sometimes people are lactose intolerant, and sometimes that makes them have responses through their stomach, so such as vomiting or diarrhea. Allergic responses to insect stings, so like those that are allergic to bees, sometimes we see a swelling around the area that they are stung, or even a severe allergy to bees, maybe even see an anaphylactic shock where again breathing is then cut off. So things of that nature too. So those all fall in that type 1 hypersensitivity, which is your most common type. So the allergic response is that type 1 hypersensitivity, again, probably the most common type of hypersensitivity. Okay. You do not have to know the specifics of how the allergic reaction takes place. I meant to take this off of the slides here, so you guys don't worry about that. Just know that the allergic response falls in that type 1 category, and it is the most common type of hypersensitivity. And this just kind of shows the factors in the development there, and you guys don't have to worry about that. Okay, and I mentioned too, we don't have to know the specifics of what happens to create that allergic response. I'm going to skip through that. I do want you guys to be familiar with that type 1 hypersensitivity reactions could be either localized or systemic. So we've talked about that before. Localized, again, meaning that it just stays local to an area. So such as a bee sting, sometimes we just see swelling around where that bee sting occurs. So it's going to be not as severe. So they result in allergic reactions such as hay fever symptoms, asthma, food allergies sometimes can just be localized. Systemic reactions, however, you don't need to know that it's basophil degranulation, but this means that it occurs throughout the body, and these can be more life-threatening. So this is where the anaphylactic shock comes in, where maybe breathing has been cut off and it's a little bit more severe. So people with very severe allergies to certain things, where this may be a result, the anaphylactic response may be a result of their allergy, they typically are the ones carrying around those EpiPens, because epinephrine, if you guys remember from the endocrine system, epinephrine is going to jolt their body awake pretty much. It will open up the bronchial tubes, making breathing much easier. It's going to increase their heart rate tremendously, though. And so typically after somebody has been administered an EpiPen, they need to make sure they get to the hospital so that heart rate can be monitored, but it is going to at least make sure that they are breathing after being exposed to an allergen. Okay. So let's skip this. You guys don't, I just wanted you guys to know that systemic, allergies can be a little bit more life-threatening. This just gets into, I always leave this here for your alls if you all want to, as those that again experience allergy skin testing and allergy shots. This can be tested to identify, what pathogens or allergens a patient is allergic to. If anybody's been through those skin tests, you guys may be familiar with that and know how not pleasant they are. But that's, again, like I said, we're not gonna worry about that. So I'm gonna skip through those. We're gonna skip through type 2 sensitivity and type 3. I meant to delete those off there. And the only thing I want you guys to be familiar with type 4 is that it is the delayed type, like I mentioned earlier. So meaning we're seeing results, or not results, but a response. So that hypersensitivity response occurs 24 to 48 hours after contact. Okay. And then autoimmune diseases. So I'm going to be brief with this as well. So autoimmune diseases. So finishing up our immune system here, autoimmune diseases result when a person's immune system can no longer recognize body tissues as self versus non-self. So we mentioned at the very beginning of this chapter that that's our immune system's job. We know what belongs and what doesn't, and then we destroy what doesn't. So what happens with somebody with an autoimmune disease, they can't tell the difference and they destroy both. They say, I don't know, so I'm just going to destroy both of these, and start destroying their own healthy cells. So this may occur with certain tissues that are not exposed to the immune system during fetal development. And so therefore they're not recognized as self because it doesn't recognize that. It wasn't exposed to that in that development stage. There are more than 80 recognized autoimmune diseases. So there's lots of different types of autoimmune diseases. So they can be classified as either organ specific or non-organ specific. And there's a couple examples down there. Graves' disease, lupus is one. So there's just different examples that you guys may have heard of. That's all I want you guys to be familiar with autoimmune disease, what it is. And again, it's pretty much the immune system not working properly. Okay, we're going to skip through immunosuppression. So go ahead and skip through the rest of this, and then that will end the chapter there. Sorry, I meant to cut up that PowerPoint slide a little bit so that we can make sure we focus on just the things I want you guys to be familiar with. So we'll end with the autoimmune diseases there. All right, guys, if you all have any questions whatsoever, you guys just let me know.